Welcome to part 2 of my lecture about molecular mechanisms of HPV-induced cancer. So in this part, I will talk about HPV infection and how HPV can induce cancer. So here we see cervical carcinoma in situ in the middle of the picture, and we can see also different causes of cancer like molecules, chemicals, radiation, inherited cancer genes, and viruses, and HPV. So when HPV infection occurs, usually the HPV infection is cleared after 1 year, and 90% of all HPV infections are actually cleared after 5 years. But sometimes the HPV infection become persistent, and then cells are transformed into neoplasia. First, they transform into lower grade squamous intraepithelial neoplasias, but after about 5 years, some of these low grade lesions can become higher grade lesions. And if the HPV infection is persistent and the high grade lesions persist, after some more years, at least 10 years or more, these lesions can transform into cervical cancer. So why does this happen? I will talk about how this can happen, this transformation, how this can be caused by the HPV. So first I would like to show you the HPV genome. The HPV genome contains 8,000 base pairs, and these 8,000 base pairs code for several different genes. We have early onset genes, they are named E1 to E7, and we have some late onset genes, L1 and L2. So E1 and E2 are the first genes that are transcribed when HPV is infecting human cells. These E1 and E2 genes, they are involved in the replication of HPV episomes. E4 is believed to be involved in viral release. And then we have E5, which is involved in cell proliferation and maybe also immune evasion. And E6 and E7, these genes are involved in the maintenance of HPV episomes and the cell cycle control. And then the late onset genes, the L1 and L2, codes for the capsid proteins. So here we see a picture of the HPV life cycle within the human cells. So first, the HPV, I will show you here. The HPV particle needs to enter the basal cells of epithelium. So there has to be some kind of scratch in the epithelium so that the HPV particle can infect the basal cells. And when this infection occurs, the genome maintenance genes are being transcribed, E1 and E2. And then the virus episomes are being replicated. So the HPV DNA is replicated within the episomes, and the genes that are involved in genome maintenance and cell proliferation are now transcribed. In the upper layers, also gene E4 starts to be transcribed, which is probably involved in viral release. And in the upper parts here, in the blue layer, the cornified layer, the late onset genes L1 and L2 is now also being transcribed, and those proteins are, and the genes, the RNA is translated into the L1 and L2 proteins, and the virus is then packed. And assembly into particles. And these particles can now be released and infect other cells, other neighboring cells or other humans. So the HPV can continue to spread. So when we have HPV and gene P53. So in HPV-associated cancers like cervical cancer, if you remember this table from the lecture, the part 1 of the lecture, cervical cancer has only, it's quite few cervical cancers that have mutated P53 gene. Only 6% of the cervical cancers. So, and this is seen in HPV-associated cancers, that these cancers have intact P53 genes and they have normal TP53 transcription. However, there are no detectable P53 protein in the cells. So how come? It is about the E6 gene from the HPV genome. Because the E6 protein that is translated from the HPV genome, the E6 protein can bind to P53 together with an associated protein called AP. And when E6 and AP bind to P53, it will degrade P53. And this leads then to the cells, they will be resistant to apoptosis, as P53 can cause apoptosis. But cells that express a lot of E6 will become resistant to apoptosis. And this also leads to an increased chromosomal instability within those cells. We have also the HPV gene E7. This E7 protein can bind to the tumor suppressor protein retinoblastoma, RB. And when the E7 binds to RB, it blocks the RB to bind to the transcription factor E2F. And if you remember from the lecture part 1 here, the transcription factor E2F is a transcription factor for the S-phase genes, genes that are necessary for the cell to be in the synthesis phase and to continue cell proliferation. So when the E7 is expressed in the cell, it binds to the RB protein and the RB is inactivated. So we will have free transcription factor E2F in the cells. And that leads to that the S-phase genes are transcribed and the cell cycle will progress. So here we see the complete picture of how E6 and E7 are oncogenes in the high-risk HPV types and how they act. Both the E6 act on the P53 pathway by degrade the P53 and then stopping cells from apoptosis, and how the E7 protein from the HPV can bind to the RB protein, leading to cell proliferation. But why are these E6 and E7 oncogenes in some HPV types? What happens here is that, first of all, in the beginning, when we have a new HPV infection, we have also the HPV gene E2. And E2 protein regulates the transcription of E6 and E7. But when the infection of HPV becomes persistent and it stays within the host cells for a very long time, eventually the HPV genome can be integrated into the host genome, into the human genome. And when this integration occurs, the circular HPV genome of 8,000 base pairs becomes linear and is integrated into the human DNA. However, when this occurs, usually parts of the HPV genomes are lost. And quite often, the part that is lost is the gene E2. that regulates the E6 and E7 transcription. So the result will be that there will be an increased E6 and E7 expression when HPV genome becomes integrated. And if the cells then will contain a lot of E6 and E7 proteins, these E6 and E7 proteins will bind to the P53 and RB proteins, and the cells becomes resistant to apoptosis. The cells will have an increased chromosome instability and increased cell proliferation, and this in turn leads to cancer. So here we see a study by Durbar et al. Published in 2012. Here we see HPV E7 expression in cervical cancer. So if we just look at the left picture here, we see different grades of cervical neoplasia. So to the left here, we have some uninfected normal cells. Then we have some low-grade lesions, higher-grade lesions, and then more high-grade lesions of cervical epithelial cells. And the right picture here, now the cells are marked with two different markers. The red marker is a marker for E7 expression. And what we can see here that E7 is not expressed at all in the normal cells, which was expected. They are expressed at moderate level in the SIN1 grade lesions, but they are even more or higher expressed in higher grade lesions, like SIN2 and SIN3 lesions here. The green marker, however, is a marker for E4 protein. And what we see here, the interesting thing here is that we have high expression in lower grade neoplasia. However, in the high grade neoplasia, where we have cervical cancer in situ, or SIN3, the E4 expression is actually lost. And this is also fits well with the hypothesis, or with the hypothesis here that the HPV genome can actually be integrated and parts of the genome then is lost. And E4 gene is within E2 gene, which is usually lost when the HPV genome is integrated. And this picture here shows how E4 expression also is lost in the higher grade lesions. So what we have when we have an HPV infection causing cancer is that we can see a change in the HPV gene expression when we look at different grades of neoplasia. So in low grade neoplasias, SIN1, we have expression of both E7 and E4, as well as the late onset gene L1. And we also have a very high number of virions that are released on the surface that can infect other cells. With higher grade lesions, we have a reduced number of virions released actually at the surface. We have also higher levels of E7 and but E4 is at the same level or a bit less. And when we have cervical cancer, there are no more virions released, and the HPV genome is only expressing, not expressing E4 anymore, but it still expresses a lot of E7. But now HPV episomes are also lost. So now the HPV genome is only contained within the human genome, as integrated into the human genome. But why does the human papillomaviruses cause cancer if there are no more virions that are released? The cancer is then an end for the HPV infection. It cannot spread anymore. So why did these high-risk HPV types develop and become cancerogenic or oncogenic? if this is not good for the virus spread. So we can have a look at the HPV from an evolutionary perspective. Here we see the HPV tree. So different mammals have their own papilloma viruses. And we see the green ones at the top here are some human papilloma viruses. And what happens during the evolution is that the E6 and E7 gene functions, they evolve during this evolution. And some HPV types become high-risk types because they have more oncogenic E6 and E7 genes. And the E6 and E7, they increase cell proliferation, and they may also prevent apoptosis. But cancer, as I showed you, cancer does not help the virus to spread. So there is no evolutionary advantage for the virus to become oncogenic. So why did those high-risk types evolve? The time is the important factor here. As you remember, for an HPV infection to cause cancer, it takes at least 10 years. The median time is 20 years from the initial HPV infection until the cancer has developed. So during this time of 20 years, the virus has a very long duration of spread before the cells has turned into cancer and the virus cannot spread anymore. So it's a matter of time. So the advantage is that those high-risk types, like HPV-16, has longer time to clear than other types. And this leads to a more efficient spread as they stay a long time within the host cell. So it has a longer duration of infectivity. However, the drawback of this is that if they have more efficient spread, it is also more oncogenic, because the reason that they can stay long within the host is that they are more oncogenic. So for a take-home message from this part 2 lecture about HPV and cancer is that HPV infects the basal cells of the epithelium, and the HPV infection can become persistent. And the HPV genes E6 and E7, these are oncogenes in some of the HPV types. And these genes can prevent apoptosis and increase the cell proliferation. And infection with oncogenic HPV types, they take longer